So hello, everyone. So I'm Arthur, and I work on developing dimensionality reduction methods for large-scale neural data, and also some neural network modeling. And today I'm going to tell you about dimensionality reduction methods. So we are in this great age of neuroscience where we have access to increasingly high-dimensional neural recordings. We can now record from tens of thousands of neurons in behaving animals across long timescales. But you have this data, what do you do with it? You have this matrix that's mentioned 10,000 by a couple hundreds. How can you tell something out of this data? That's what we are going to try to answer today. And before we tackle this somewhat difficult question, we'll try to answer something that's much simpler, namely how we can say something and do dimensionality reduction methods on just a tiny subset of this data that's just this tiny piece of this matrix, in particular, just the activity of two neurons. Okay, so we have this data matrix that contains the activity of these two neurons across time. And one perspective that's been particularly helpful towards understanding neural data is that of the state space, where we can define a vector space where each axis corresponds to the activity of one neuron. And then within this space, my data can be represented as data points. More specifically, if I take one of my data points, so the activity of my two neurons at a particular time point, this will correspond to a data point within this state space, where each coordinate of this point corresponds to the activity of one of the neurons. And what do I mean by the activity of a neuron? Well, that depends. If you are working with calcium imaging, it could be calcium signals. If you are working with spikes, it could be beam spikes. But at least as far as the dimensionality reduction method is concerned, we are just using this state space view. Okay, so what's the next step? Well, we want to do dimensionality reduction. And in particular, here we have access to this two-dimensional state space, and we want to reduce the dimensionality. So we are going to reduce the dimensionality onto a one-dimensional space, namely just onto a line. And the way we are going to do that is that we are going to project the data onto this line. And that's what we will call doing dimensionality reduction. Now, what is this one-dimensional subspace? What is this line? Well, that depends on what method you use. So for example, if you want to maximize the variance of the data onto this subspace, then you will find this subspace using PCA. If you want the sparsest representation possible, you will use SCA. And if you want your subspace to be strongly correlated with task variables, maybe you will use targeted dimensionality reduction. But in all of these cases, we are doing the same thing. We are finding a low-dimensional subspace and just projecting the data onto this subspace. So moving forward, we'll kind of abstract what method we are using. Okay, so we have this line, we've projected the data on it, and now we want to view the data within this line directly. So move away from this state space. And for this, we are going to do this dimensionality reduction step, where we are going to describe the data in terms of the coordinates that it has on this line. And that's going to be our dimensionality reduction step. And the way we can do that is via a matrix that is just going to take different linear combinations of the activity of my two neurons to characterize this latent variable. So now we have a low-dimensional description of the data, so we can just visualize the data within this subspace. Okay. So I think so far we've been pretty explicit about everything that's going on. So far, you should have understand how things work. And what I'm claiming is that if you understand what I just explained, then you can understand the 10,000 dimensional data. So let's go back to it. So we have this 10,000 dimensional matrix. And just like we defined a two-dimensional state space when we had two neurons, we can define a 10,000 dimensional state space for these 10,000 neurons. And this state space is so high dimensional that there is really no way to visualize it. We cannot picture in our head of a 10,000 dimensional space, but at least we can make this little schematic on the right to kind of keep track of what's going on. Then if we look at the activity of our neurons at a particular time point, that will correspond to a vector or a point within this state space. And as time progresses forward, the activity will change. So the coordinate of this point will change. And overall, this will describe a neural trajectory. And what people have found empirically is that these neural trajectories, they are often constrained to low-dimensional subspaces or low-dimensional neural manifolds. So now, just as we did for the two-dimensional example, where we projected it on a one-dimensional line, we can project this 10,000 dimensional data onto this two-dimensional plane. And we can visualize the data directly within this plane. And that's how it looks like. And what people have found is that within these low-dimensional subspaces, different coordinates, different axes within the space are often correlated with behavioral or task variables. So for example, here, different points correspond to the activity of the neurons at different time points, and the different colors correspond to different values of the task variable that the animal is currently experiencing. And this view of studying neural data from this geometric perspective, from this low-dimensional subspace perspective, has been really ubiquitous in neuroscience. One of the earliest example that I know of is in the locust, where they looked at the olfactory system of this insect and looked at the representation of different odors. They had recordings from tens or so neurons, and then they presented the animal with different odors, which here are represented by red, green, and blue colors. And they presented these different odors at different concentration levels, different strength of the smell. And so just as before, they had this high-dimensional data, and they projected it to a low-dimensional subspace. And in particular, here, they chose a three-dimensional space. And what they found was that within this three-dimensional space, the neural activity seemed to cluster the different odors and to capture something about the continuity of the concentration gradient. And since then, people have really applied the same idea to several different types of data and several different types of tasks. So for example, at some point we had access to the recording of the same neurons during behavioral tasks using data arrays, and people applied these dimensional introduction methods to these motor data. And then more recently, people have been doing exactly the same thing on calcium data. And the key takeaway from this slide is that across all these examples, we are doing the same thing. We are taking this high-dimensional data and projecting it onto a low-dimensional space, and just color coding the neural activity according to the task variable. And in all of these cases, we find that the subspace captures something about the task structure. Okay. So when does that not work? A classic example of this is that of neural sequences. And in particular, here I'm showing data from the songbird. So there is this particular nucleus in the brain of the songbird that's related to producing songs, vocalizations. And what they observed was that in this particular nucleus, the activation of the different neurons was sequential. So if you look at the data matrix, one neuron is activated, and then the next, and then the next, and then the next. So it makes a sequence of activation. And when we visualize that from the point of view of the state space, that's how it looks like. At different time points, the population vector, the point within the state space, is aligned with individual axes of the state space, the one corresponding to the neuron that's active at this particular time point. And because this population vector is aligned with different axes at different time point, it means that the neural activity is not well constrained to a low-dimensional space. or said differently, we would need a subspace that's the same dimensionality as the neural data to represent the data. And these sequences, they are also very ubiquitous. So people have found them across different species, across different tasks, and across different brain regions. And so that kind of hints at the fact that perhaps we should kind of depart or extend our neural subspace view to capture more types of structure in neural data. And one way people have been doing this is by extending this low-dimensional subspace view to nonlinear manifolds. So instead of representing the data onto a low-dimensional subspace, represent the data onto a nonlinear manifold, curved manifold. And in this particular work from 1 year ago, what they did was on these kinds of data where monkeys are doing reaches, they're recording from the motor cortex. They looked at how much variance, or conversely, what was the reconstruction error of linear versus nonlinear manifolds across different dimensions of the manifold. So here the horizontal axis of the plot on the right corresponds to the dimension of the manifold, so line, plane, volume, and so on. And the vertical axis corresponds to the reconstruction error of the data using this particular dimension across the nonlinear and linear manifold. And what they found was that the nonlinear manifold was capturing the data with many fewer dimensions. And so these nonlinear manifold discovery methods have been applied to many different types of data. So for example, in the head direction system, there are cells that represent where the animal is facing. So here they applied the dimensional reduction method that I won't describe, but some nonlinear dimensional reduction, and then color-coded the points according to where the animal was heading, and they found that the circle was encoding the direction of the animal. And people have also found toroidal manifolds in grid cells that encode the position of the animal in physical space. So here in the little inset, you are looking at the position of an animal per time in physical space, and that corresponds to a trajectory on the stars in the neural manifold. So these nonlinear manifolds, they are good. They can cover some relevant structure in the data, but there is a caveat. So in going to these nonlinear manifold methods, we've kind of lost interpretability. In the linear case, I was able to fully explain what was going on in the first slide by showing you the subspace, showing you the matrix that did the projection, explaining that it maximized the variance or the sparsity and so on. But in the case of these nonlinear manifold methods, we've kind of lost that. The method is much more complicated than we can explain in a simple manner. Okay, so I'm going to spend the remainder of this talk to try to address this. And in particular, what I'll argue is that linear methods can do as well as nonlinear methods if we just consider or view the data slightly differently, or if we choose the right view of the data. And for this, we'll need to go from data matrices to data tensors. So far, I've described how to do dimensional reduction on a data matrix. But in neuroscience experiments, we are not just sampling a single data matrix. We're repeating the same experiment over and over, and we have data over different trials. And then what we can do is perhaps trim or warp this data such that they are of the same duration time, and then represent this data as a tensor. So here, each slice of this tensor, this multidimensional array, corresponds to one particular trial. And then we can do this kind of weird step at first of unfolding the tensor back into a matrix, but doing that in different ways. So we can either unfold this tensor into a matrix that's of shape number of neurons, some very long dimension that's number of time points by number of trials, or unfold it into a matrix that's number of trials by a very long dimension that's number of known by number of time points. And what people have argued is that these different views of the same data can capture more or less variance depending on the brain region that you are looking at. So here I'm showing the reconstruction error of applying a linear dimensional reduction method to either unfoldings of the data in the visual cortex and the motor cortex. And we can see that in the visual cortex, the top matrix is better. It has a lower reconstruction error. In the motor cortex, it's the opposite. So why does that matter? Well, because it can capture more behaviorally relevant information. And to illustrate this, I'll show data from, again, a reaching task. So where a monkey is starting at the center of the screen with its arm and tries to move towards a particular target. And there can be a maze or no maze. And if there is a maze, the monkey has to go around it. And the first thing that we can do is just average the data within each condition, so within each target location, to get this data tensor, and then just regress the neural activity to the behavioral variable. So regress the neural activity to the position of the hand of the animal. And if we do that, then it's great. We can decode the position of the hand of the animal. However, if we try to do that on the raw data, it doesn't look as good. The decoding is not as good. So it looks like there is variability in the data that's perhaps relevant for behavioral information that's in the raw data, but there is also variability that's just not related to the behavior. And so one thing that people commonly do to get rid of this noise or unwanted variability is to do PCA on the neural activity, which is to apply a dimensional reduction method to this matrix that we saw earlier. And if we do that, and then apply a regression method to decode the position of the hand of the animal, it does very poorly. But surprisingly, if you do the same thing, but on the other representation of the data, then it does really well. In fact, it does as well as nonlinear methods. So for example, here on the right, I'm showing LFADS, which is a fully nonlinear method based on neural networks. And just applying linear dimensional reduction methods, but on the right representation of the data does just as well as this nonlinear method. So I'll end by mentioning, again, this point that different brain regions might prefer different sizes, different representations of the data. And for this, I'll briefly mention a NeuroPixel data set. So it's this IBL data where mouse is moving gratings from the side of a screen to the center of a screen, and they're recording from a couple of brain regions with NeuroPixels. And what we found in our work is that different directions in the state space uncovered by linear dimensional reduction methods were capturing different brain regions. But perhaps more interestingly, if we represented the data in the other way we can represent the data, then it captured something else, something about correct versus incorrect choices of the animal. Okay, so at this point, you might have technical questions or concerns. So how do we choose the dimension of the subspace? How are you guaranteed to always find the same subspace and so on? And if you have questions related to this, I invite you to look at our paper where we address these technical types of questions. So in summary, dimensional reduction works by projecting high dimensional data onto a low dimensional space and visualizing it. But we need to be careful choosing the right representation of the data. And if we do so, then these linear dimensional reduction methods can perform just as well as fully nonlinear methods. And also different brain regions and also different kind of task variables might prefer different views of the data. Thank you.